All right, we're up to almost 20 people, so I will kick us off. So first of all, welcome everyone. Thanks for joining today. For anyone that's attending this call for the first time, we've been holding this industry-wide call every month with companies that essentially want to contribute to the cultivated meat value chain. And so the purpose of these calls is really to offer new and existing cultivated meat stakeholders an opportunity to learn from each other, especially with respect to some of the obstacles that you're facing and some of the potential solutions that might exist along different aspects of the value chain. Ultimately, we hope that this will help accelerate some of the R&D and commercialization in the sector and form new collaborations amongst different stakeholders. So if your group is interested in presenting on a future call, maybe you're working with a company that could benefit from being on these calls, please do feel free to put them in touch with me directly. I'm happy to send them an invitation. Also, a reminder that you are welcome to extend this invitation to colleagues at your company as well, in case they're interested in attending calls, keeping up to date with presenters, or hearing announcements from GFI. Just one housekeeping item from our side today. So just wanted to give folks a heads up that if you are someone that's been working on cultivated seafood, we have a survey that is going to be sent out shortly in the coming days. This survey is specifically structured to help us understand challenges related to immortalization of cells from aquatic animals, which we know has been challenging. And so this project's being led by my colleague Claire at GFI, and we hope that really taking the collective insights from people, their learnings over time will help really drive progress forward in a faster rate in the cultivated seafood field. So please be on the lookout for that survey in your inboxes, and appreciate you taking any time to share your experiences, especially if you have direct hands-on experience with isolating and immortalizing cell lines from aquatic animals. So on the topic of cell lines, today I'm pleased to be welcoming Ramiro Alberio from the University of Nottingham. He's going to be discussing development of some animal pluripotent stem cell lines, and they'll be sort of co-presenting with Dylan Stavish from STEMCELL Technologies, who's going to be diving a little bit deeper into the development of suitable media for porcine pluripotent stem cells in particular. So as always, feel free to drop questions in the chat throughout, and hopefully we can get to them at the end of the presentation. Ramiro, I'll turn it over to you.
Great. Thanks, Elliot, for the introduction and for the opportunity to present at this forum today. As you just alluded to, so we're going to have a double act today. So it's going to be me introducing some of the underpinning research that has led to this collaboration that we have developed with STEMCELL Technologies. And Dylan will be joining me in the second part of the talk to describe the collaboration that we established and the product that we developed together as a result of this joint program of research. So just to give you a brief overview of our lab. So we are a lab that works on embryogenesis in livestock species. And as part of our research program, we developed in the past 8 or 9 years novel embryonic stem cell lines from sheep, cattle, and pig embryos. And these cell lines are very much amenable to differentiation into multiple lineages. And we have basically spent quite a bit of time characterizing these cells for applications such as cultivated meat. So what I would like to give you today is a bit of an introduction as to what the features of these cells are, what makes them a bit different to alternative cell lines available for this industry, and maybe have a bit of a discussion as to why the collaboration with STEMCELL Technologies came about, and addressing particular type of bottlenecks in this area. So I'm based here in Nottingham, and as I said, I've been working on stem cells for many, many years. And for those of you who are not familiar with the sources of cells used for cultivated meat, this is a picture taken from a recent paper published by a European group where they summarize basically the different sources of cells that are currently available. And it is a very, it seems like a very complex slide, but I basically divided this into 3 major groups of cells that can be used for cultivated meat. And the major group of cells currently used are tissue biopsies, where people obtain either muscle cells from the satellite cells of the muscle, or adipose tissue that can be expanded in the lab to great amounts, and then subsequently be used for trilineage differentiation, depending if they are mesenchymal cells or satellite cells, to generate muscle or fat cells. This is where the majority of the industry is based on, using this type of samples. There is also the alternative option of using embryo-derived stem cells, and these are the ones that I'm presenting here today. So these are cells that are obtained from a pre-implantation embryo, and these cells can be expanded in the lab and maintained very long terms, very long time, and used to differentiate in the desired tissues or cell types. And so in this case, we focus on making muscle and fat, and we'll show you data on this in a moment. The third type of cell that is maybe less developed, but there is a lot of potential for this type of cell, is a cell obtained from a skin biopsy from an adult animal, for example, which can then be induced to pluripotency using induced reprogramming technologies, and subsequently can be induced to differentiate into muscle tissues. A lot of research is going into this topic, but is, as I say, is less developed than the other two areas. So I'm going to be focusing primarily on the blue side of this graph, and just to give you some technical details about these cells. So these cells have only become available recently, because they were derived around 2015, 2016, and very few papers really describe this type of cell from previous studies. The advantage of the derivation that we achieved in these experiments is that we obtained cells in animal-free conditions. Previous experiments had dealt in the derivation of stem cells from animals using feeder layers and a number of animal-derived products to support the growth of the cells, including serum and other components. But the key achievement in these experiments here was that we were able to derive this in chemically defined animal-free conditions. And this was the first time this was done. And this allowed us not only to have a suitable product for food consumption, for example, because there were no animal products involved, but also to define the requirements of the cell very carefully. And so we were able to establish that a set of growth factors and inhibitors were required for maintaining these cells in a long-term serving unit capacity. And these factors are FGF, activin, and Wnt inhibition. With these conditions, we were able to characterize the cells very thoroughly and establish that they can grow pretty much indefinitely in these conditions when they are finely tuned to the right levels of FGF, activin, and Wnt inhibition. And here are examples of the type of markers that we normally use to characterize the cells. You can see the expression of these three genes, OX2, NANOG, and OX4, are the classic markers of pluripotency. They are expressed at high levels in all these colonies of cells. We can also use FACS sorting to purify the cells, and we obtain high enrichment for these cells. And alkaline phosphatase, which is a classic marker of pluripotency. So once you start removing the different factors, we know that if we remove FGF, for example, in this AX condition, we lose pluripotency. When we block MAP kinase signaling, we obtain a similar negative result, meaning that FGF is acting via MAP kinase, as well as the removal of Wnt inhibition and activin have negative effects. So we are very we're able to define the three critical components defining the pluripotent state in these cells. And when we align these cells using genomic technologies to the developing embryo to identify which part of the embryo they align most closely to, we identify this period between day 8 and day 11, which in a pig is around the time when the blastocyst is about to undergo the next process of development called gastrulation. So that means that these cells very much represent the cells that are in the embryo very early in development, before any cell fate decision has occurred. So with this in mind, we decided this offers an opportunity of using these cells for technological applications, and clearly cultivated meat became an option. And so we decided to differentiate these cells into the muscle lineage. I think this is a video. I'm not sure it's able to play, but oh, there we go. So this shows, for example, the differentiation of these cells into multinucleated muscle cells. You can see here the twitching of this cell. This is a little movie that we made of these cells. And when we look at different markers for differentiation, we see that these cells can differentiate the fully differentiated muscle cells. You can see titin here is a marker of mature muscle, and also myogenin. So these cells have the potential to contribute to muscle in the right conditions quite efficiently. Similar experiments were done for fat, for example. These are in the paper. Just for brevity, I just focus on the muscle side. The important thing is that these conditions that we developed can also be used in other livestock species like sheep and cattle, and also in rodents and humans. So we repeated the experiments using these similar conditions in all these species, and similar cell lines were established, which means that this is a very kind of a core cocktail that allows the capturing of the pluripotent state across multiple species. And this also offers interesting flexibility and application to, obviously, in multiple species. So the point I would like to stress about the use of pluripotent stem cells, and maybe to have a bit of a discussion as to why we think this is a desired cell type and useful cell type for the cultivated meat sector, is because it ticks a lot of boxes with regards to some of the criteria that are considered as highly desired or by companies, particularly that want to make this process very efficient. So if we look at, for example, traceability, which is an important aspect for consumers and the regulator, we have very good traceability from embryonic stem cells. These embryos can be produced and be identified very well from the donors, which animals are the donors and the source of these animals. There is no use of antibiotics at any time, so these cells are extracted from this blastocyst and are never exposed to antibiotics. They have high differentiation potential, as I showed, and they have very rapid proliferation. So these cells are programmed in the embryo to divide on a regular basis. Typically, we have a cycle of 16 to 20 hours, depending on the cell line, but it's relatively rapid, which compared to other cell types, sometimes somatic cells can be quite slow at their division, and they have also limited potential for long-term proliferation, as we know. And that's why there is a requirement for immortalization in some cases in somatic cells. In this case, these cells don't require immortalization. They're naturally immortal because they are programmed to divide. And then when taken out of the embryo, maintained in the right conditions, they will divide basically forever. They have genetic stability that is proven by karyotypic analysis, and this is very much dependent on how the cells are grown. And so we believe that in the conditions that we grow in the cells can maintain high chromosomal stability. And it's highly reproducible. We have multiple cell lines obtained from multiple species, and pretty much every time you put an embryo through this process, you will, if the embryo is good quality, you will get a cell line at the other end. So it's a very reproducible. And from the point of view of manufacturing, this is very important to have something that can be, every time one gets an embryo, one can get the cell line at a very high rate. The efficiency is very high, and again, these cell lines are all commercially available. So that makes, from a manufacturing point of view, a very interestingly accessible product to work with. So as a result of that, we created a platform called PluriCells, which basically offers these cell lines to the industry. These cell lines are food compliant. They are available from different species. They are very well characterized, and they are provided through this platform to academic collaborators around the world, which are using it to develop different protocols for downstream processing, from scale-up to differentiation methods. And they also offer to industry. So multiple companies are using our cell lines for developing their platforms. So as part of our quest to develop the supply chain and provide a high quality product for the industry, we've been identifying some of the critical needs in this area and linked with specific partners to develop specific products and solutions to this. And so one of the major cost relating to cultivated meat is related to production costs, particularly linked to culture medium. So when we looked at the entire process that entails our part of the process, which is creating the cell lines and stocking up and scaling up the number of cells, we found it was important to address the cost issue of medium, which was the primary cost in our production system. And so we partnered with STEMCELL Technologies to see if we could develop a novel method for growing these cells that would bring this cost down substantially and improve the number of cells that we could obtain in every single experiment. And so this is the collaboration that we started about a year ago with Dylan and colleagues at STEMCELL Technologies. And this is the part that I would like now to pass on to Dylan to complete and talk to explain the product that we co-developed between the two labs. Before I finish, I'd like just to briefly mention the people that were involved in my lab. So this is a cellular agriculture lab in Nottingham, which is made of Doris, Nathan, Chen Li, Charlotte, and Luke. And again, much of the work that we develop in Nottingham is funded by our research council funding, as well as collaboration with STEMCELL Technology and Avifarm Technologies, a cultivated meat company that works down in Oxford, with whom we have a strong collaboration. So perhaps I now pass on to Dylan to continue the talk, and maybe we can open this for questions at the end. Thank you.
Thank you, Ramiro, for that introduction. It helps me with the background. And thank you for the Good Food Institute for the invitation to actually present some of our work. So my name is Dylan Stavish. I'm a senior scientist at STEMCELL Technologies, working in the pluripotent stem cell biology department. And today we're going to give a brief conversation about our work on making a robust maintenance media for the culture of pluripotent stem cells from porcine. So just a little background of the company. STEMCELL Technologies is not that young. It's 30 years old now, and is based out of Vancouver in Canada. But you can see here that kind of have a global network now. And myself, I'm actually based in the United Kingdom and work out of our research institute in Cambridge. So we're kind of spread everywhere, and we've got our fingers in many pies. And we have a lot of product lines and research avenues that we do a lot of collaboration and investigation in different biological aspects. In particular, relevant to this is our work in the pluripotent stem cell biology field. Now, as Ramiro sort of said, he kind of came to us with this collaboration and knew about our extensive knowledge in growing human pluripotent stem cells. And we have a particular lineup of HPSC maintenance media, such as MTESR1, MTESR Plus, that have different compliancy levels and different applications that could be useful for us. And initially, what we sought to do was to see, do any of these medias actually work on porcine cells as they are? So after we got some of the cells, we threw them in the different conditions. But what we found is that in the standard culture conditions, the porcine PSCs really failed to thrive. And there was very little that we could do to maintain them by just using the standard mediums. One of the medias that did stand out in this early testing was that of eTeaser, which is a newer medium from our company. And eTeaser was developed initially to support human PSC expansion, and it contains optimized components that support HPSC growth and expansion to reduce selective pressures associated with single cell passaging. And we thought that the idea of single cell passaging might be useful in terms of scale up in the cultivated meat space, because handling single cells allows for automation much more readily and more reproducible growth dynamics, because you're able to count and see the same amount of cells. So we started thinking about using eTeaser as a base. And E-teaser has been shown in human to, retain those classic characteristics of PSCs, such as this long-term expansion and the gene and marker expression that you would associate with pluripotency. And another key aspect of it is that these expanded cultures actually in E-teaser acquire fewer cytogenetic changes or mutations than current commercial cell culture media when passaged as single cells. So it offers a greater genetic stability to these pluripotent cells, which if you're planning on scaling and growing and growing and growing, you will need to maintain a good karyotype in the cells. And kind of through this collaboration, we came across the same thing as Ramiro pointed out, that activin A and ZAV939, this Wnt inhibition, is very critical for the supporting of porcine PSCs. And when we actually added those two small components into E-teaser media, we found that we got very nice porcine PSC growth. When we actually compare that to the published formulation that Ramiro was speaking about, N2B27 with AFX and E-teaser plus AX, we find that we get a much nicer colony morphology. We get slightly tighter, more compact colonies, a little bit less differentiation towards the edges, and all around more morphology that you expect to see from human pluripotent stem cells, actually. So we saw very nice growth images down the microscope, and so we were very confident that we might have something working there, and we decided to take that out further. So we actually were able to maintain the porcine PSCs in this E-teaser with AX media, and also the published formulation. We did it across 4 lines for 75 days, and we saw enhanced growth rates in the E-teaser and AX over the published formulation. And you can see that kind of here, our average 5-fold day expansion for some of these lines. In the E-teaser formulation, we're seeing up to 425-fold increase in the expansion, compared to the standard publication was only 92. So we're seeing much greater fold expansion of these cells in E-teaser and AX. And that sort of equates to an increase, 82-fold increase across the 4 cell lines. This was very consistent for us, and this is just sort of breaking down the by passage compared published and E-teaser, and it's very consistent that we see this enhanced growth rate in the E-teaser and AX across all the cell lines, and that was a significant difference for us. And we also validated that by looking for some of the characteristic undifferentiated markers that Ramiro pointed out, such as OCT4 and SOX2, and we were happy to find that our OCT4 and SOX2 actually are designed for human, but react for the porcine, so that was a nice useful tool for us to characterize these cells further and show co-expression of these 2 markers in our cells. And when we take those cells and we expand them for 10 weeks, these 75 days, and we actually compare the E-teaser and the published formulation, we can see that the E-teaser AX had higher levels of OCT4 and SOX2, double positive cells, across the 4 cell lines. And another undifferentiated associated marker, SCCA3, which is often shown to show how fit your pluripotent stem cells are, showed much higher expression in E-teaser AX at the end of 10 weeks than in the published formulation. So obviously we're seeing these enhanced growth rates, and maybe we want to think about how that might relate to scale of these cells. So here's just a theoretical example of how you might do a 2D scale up. And for this model, we've looked at a 40 layer cell stack. And in the blue line is if you were to grow them in E-teaser and AX, and in this beige line is the published formulation. So imagine that you started in a 6-well plate with just 5,000 cells per well. How long would it take you to grow that up into this complement 40 cell stack? Well, if you used eTeaser AX, you might be able to reach confluency in 10.7 days, whereas in the published formulation, that would take about 14.6 days to reach confluency. So that's about 4 days less time. At this particular stage, at day 10.7, you actually have about 12 billion more cells in the eTeaser AX class than you do in the published formulation flask. And by the end point of this confluence level, you actually have about 25 liters less media usage. So eTeaser is made with stable components, allowing flexible feeding schedules and miss days and skip days. So you actually use a lot less media to grow up more cells. So we would estimate around 15 liters is needed in this sort of experiment, whereas the published formulation would require 40 liters. Now, obviously, 2D scale-up is nice to visualize, but we know that many people are moving into the realms of 3D scale-up, and we decided to see how our media might perform in a 3D system. So this is just some very preliminary work using 6-volt plates, an orbital shaker. And what we found initially is that when we use just eTeaser with AX added, we get some aggregate formation and a little bit of cell growth, but it wasn't really optimal. And we found that we had to make some changes to our formulation to make a 3D optimized one. And we were able to get much bigger and better aggregate formation. And the cells were a lot happier and healthier and more uniform. You can see that here in these microscope images. We validated the expression of OCT4 and SOX2 in these 3D samples, as well as the ability to passage them on further. So just to give a summary, a kind of both mine and Ramiro stuff here, I just want to say that PluriCells supplies these high quality, traceable and stable porcine PSC cells. And really, what we've done here couldn't be done without those cells being readily available for us to play with. But just like kind of Ramiro found, I think in his early research is that porcine PSCs fail to thrive in standard human PSC culture conditions, and that the addition of activin A and ZAB939 is crucial for porcine PSC maintenance. What we found in our hands is that eTeaser and the addition of these two components can be used to maintain porcine PSCs long-term with enhanced growth rates and maintenance of the undifferentiated states. And we actually will soon be launching a protocol on our webpage, STEMCELL.com, detailing how to grow your porcine PSCs using eTeaser with a combination of activin A and ZAB939. And finally, we have some of this preliminary experiments that show a modification to eTeaser and activin A and ZAB allows the support of 3D suspension cultures. So if anyone has any questions now or later, please reach out for further information on the 2D or 3D platforms. And yeah, I guess Ramiro and I will field any questions now.
Excellent. Thank you so much for that presentation. Exciting progress in the pluripotent field. And yeah, folks, if you have questions, please feel free to drop them in the chat or come off mute. All right, well, Ramiro and Dylan, thanks so much for presenting. Yeah, great new resources for the field and really the foundational type of stuff that we need to, understand how to cultivate meat. Cells and media really form the bedrock of that. So yeah, hopefully other folks found interest in this, get in contact with you. And thanks again for your presentation.
Great. Thank you very much for your time, and thank you all for attending and the questions. So it was very, 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 very good session. So thank you.
Thank you very much.
Great. Thank you. Bye-bye.